So now we're going to look at the entrance of other monosaccharides into the glycolytic pathway. We don't just consume glucose in our diets. Obviously, we consume fructose, um, which actually comes from sucrose and can be found in sugar beets and sugar cane. But the hydrolysis of that sucrose yields fructose, um, also glucose, but the fructose must be converted or modified in order to enter that glycolytic pathway. So, structure fructose, and fructose can be metabolized into an intermediate of the glycolytic pathway in two ways, depending upon where that fructose is, whether it's in muscle or kidney or in the liver. But if it is in muscle or kidney tissue, then fructose 6-phosphate or fructose gets phosphorylated and enters the glycolytic pathway at step 3. Or if that fructose is in the liver, it's converted to glyceraldehyde 3-phosphate, which is further down that glycolytic pathway and enters in step 6. Galactose, which of course comes from lactose, glucose and galactose, or glucose and galactose, yep, form a molecule of lactose. And when this is hydrolyzed, we release glucose and galactose. The galactose 2 must be converted to a glycolytic intermediate, and that intermediate is glucose 6-phosphate. And then it too can enter the glycolytic pathway in step 2. Remember that glucose has two epimers. Remember that one of the hydroxyl groups is just in a different place. So galactose is a C2 epimer of glucose, and mannose is a C4 epimer. And just as an aside, patients with galactosemia lack the enzyme to convert galactose to glucose 6-phosphate. Mannose is obtained from polysaccharides in fruits. Examples are cranberries and currants. And again, mannose is a C4 epimer of glucose, and it is converted to fructose 6-phosphate before entering the glycolytic pathway, also at step 3. So all of the common hexoses enter the glycolysis pathway and are metabolized to pyruvate. So pyruvate, remember, is that 3-carbon molecule that is the ultimate product of the glycolytic pathway. And this can be converted to 3 other molecules depending upon the environment it is in. Pyruvate can be converted to acetyl-CoA, which then can feed into the pathway we're going to look at next, the citric acid cycle, or tricarboxylic acid cycle, TCA. And this is under aerobic conditions. If we have anaerobic conditions, that pyruvate is converted to lactate, and this occurs in muscle tissue. And alcohol fermentation, which occurs in yeast, pyruvate is converted to ethanol, and we'll look at all 3 of these possible products. Again, acetyl-CoA, if we're under aerobic conditions, lactate under anaerobic conditions or lack of oxygen, and fermentation by microorganisms yields ethanol. So under those aerobic conditions, and again, this is when oxygen is plentiful, pyruvate is oxidized by NAD+. Remember, this is an oxidizing agent. It gets reduced. And this occurs in the presence of coenzyme A, and the product is acetyl-CoA and a molecule of CO2. The NADH form needs O2 to be returned to NAD+. So without oxygen, no additional pyruvate can be oxidized. So there's a pathway that oxidizes NADH to return it to NAD+. And we've looked at that already. But here's another example. So under anaerobic conditions, when O2 is low and there's a bunch of NADH, so this is in its reduced form, it acts as a reducing agent, thus it is oxidized and reduces pyruvate to lactate. Okay. So here's that pyruvate molecule, and you'll notice at C2, well actually, yeah, it is C2, I guess, that double bond, C double bond O, is converted to an alcohol group here. So pyruvate is reduced to lactate, and NADH plus a proton is oxidized to NAD. And then this, of course, can re-enter the glycolysis pathway at step 6. So conversion to lactate, this occurs during strenuous exercise when ATP needs are high and there's not enough oxygen in the muscle tissue because it's not being inhaled to reoxidize NADH in the electron transport chain. So lactate is formed by anaerobic metabolism, and because we have pyruvate building up in our muscle tissue, and this results with soreness and cramping. So those tight and sore muscles that you feel during strenuous exercise come from the buildup of lactate in muscle cells. And this creates an oxygen debt. And when that vigorous activity decreases, you must take deep breaths of air to recover that oxygen and repay that debt from your strenuous exercise. So the lactate in muscles is reoxidized to pyruvate, which can be converted to acetyl-CoA, and all the effects of that buildup of lactate then are decreased and eventually resolve. This process, however, doesn't occur in the muscle tissue directly, or at least during that strenuous exercise. Some of it is converted back to the liver or diverted back to the liver to produce more glucose, and we'll talk about that later. An example that most of you, or all of you, hopefully have not actually experienced is pain felt during a heart attack. Surely you've experienced sore muscles from overexerting yourself or exerting yourself, but this is due, the pain of the heart attack is due to that increase of lactate in the heart muscles. And these are then cut off from oxygenated blood. Additionally, higher than normal blood lactate levels can indicate lung issues, congestive heart failure, or a serious infection. not just a result of overexertion or exertion. The third fate of pyruvate is fermentation. This is also an anaerobic process that converts glucose to ethanol and carbon dioxide. The most used example is yeast and other more microorganisms. So if any of you are bakers and you've made bread, at least yeast bread, you start with yeast and you give it some what? To activate it. You give it sugar and it starts bubbling and perking around and you smell that yeasty smell. Ethanol in alcoholic beverages is obtained by the fermentation of sugars in grapes, barley, malt, corn, rye, rice, or potatoes. And this is that process of converting pyruvate to ethanol. It involves two enzymes that you are responsible for. Pyruvate decarboxylase. Remember, we looked at this class of enzymes when we talked about enzymes, and decarboxyl, removal of a CO2 molecule, is the first step. And then alcohol dehydrogenase, which also involves the oxidation of NADH to NAD+, and the production then of alcohol. So this is reduced to ethanol. So here we have an aldehyde and it's reduced to an alcohol. So this is just an overview of the total yield of ATP from the three pathways that we're going to look at. We have only looked at glycolysis thus far, but we will look at the citric acid cycle and the electron transport chain, which is coupled to oxidative phosphorylation. And the sum of the ATP yield from those linked pathways is 32 ATP. So remember, in glycolysis, we net 2 ADP. We also get an additional 5 ATP from the NADH. And when pyruvate is converted to acetyl-CoA, we also gain 2 more NADHs, which is associated with 5 ATP down the line. And then acetyl-CoA enters the citric acid cycle. We get 2 GTPs that can be converted to or utilized like ATP. And then, and this is a combination of these 3 processes, 6 NADH, 2 FADH2s. Remember, these are the reduced forms of NAD plus and FAD or FAD. Look at this, 15 ATPs from 6 NADHs, 3 from the FADH2, and that sums to 32 ATP, plus 4 carbon dioxide molecules as a result of these subsequent pathways. So we already know, as I said, glycolysis yields a net of 2 ATPs and 2 NADHs. And this is the process by which, or an example of which, lays out in equation form these products and how we get to the 5 ATP from the 2 NADHs. And you'll notice we get 2.5, but that's times 2 because we have we get 2.5 ATP from 1 NADH. Second, the oxidation of 2 pyruvates yields 2 NADHs, which will give 5 ATPs, of course. So finally, the 2 acetyl-CoAs proceed through the citric acid cycle, and starting the electron transport chain and oxidative phosphorylation, we yield 10 ATPs times 2, giving us 20 ATPs, which in sum, from one glucose molecule, we get 32 ATPs. And you should remember this number. And you don't need to be concerned with all the details of these equations, but what's important is how many ATPs we recover from that process. So everyone's familiar with the fact that glucose is circulating in our blood, and the blood glucose levels are regulated by 2 hormones. One you're probably familiar with, insulin. So after we have fed or eaten, so after a meal, blood glucose levels rise, and that triggers the release of insulin, and the insulin then stimulates the delivery and passage of the glucose into the cells for metabolism. Once inside the cell, it gets phosphorylated, so it can't leave the cell. We've already looked at that. However, this one you might not be familiar with, glucagon. When blood glucose levels are low, so this is in between meals, if we need energy, then glucagon stimulates the metabolism of stored glycogen to break down into glucose molecules when we need them. And you should be familiar with these two very important hormones. Gluconeogenesis. Neo, new, genesis, synthesis. So this is the process by which our body synthesizes glucose from non-carbohydrate sources. So molecules like lactate, amino acids, or glycerol can be utilized to make glucose when we need it. This, of course, is an anabolic pathway. It costs energy, but end result is the synthesis of larger molecules from smaller ones. So here's an example of lactate being oxidized to pyruvate. Pyruvate then can enter the gluconeogenesis pathway and be converted to glucose. Gluconeogenesis, so glycolysis, catabolic pathway goes in this direction, and gluconeogenesis goes in this direction. Product here is pyruvate, which I'm just going to give as PYR. The starting molecule here is pyruvate, and end product is glucose. And of course, we started with glucose over here too. There are 3 steps within the pathway that utilize different enzymes in the gluconeogenic pathway. Okay. Remember, there are 3 major regulatory points in glycolysis, and it is at those points that gluconeogenesis uses different enzymes to reverse that process, and that increases the ability to regulate both of those processes. And this is an example of what I alluded to previously, and that was that lactate buildup in the muscle gets transported to the liver, and then converted to pyruvate and then to glucose that can be transported back to the muscle. And so remember, this occurs under anaerobic conditions when we need glucose, but we don't have the oxygen in the muscle tissue to metabolize it to pyruvate. And that ends this talk on the remainder of glycolysis and the production of ATP. We're going to look at the citric acid cycle next, followed by the electron transport chain and oxidative phosphorylation.